Welcome to my channel EG Life Science. Here we are going to discuss about the tissue cycle or a Kreb cycle. If you like my video, please share it and subscribe to my channel. In this lecture, we are going to discuss about the tissue cycle, its definition, introduction, the reactions occurring in a tissue cycle, and significance of a tissue cycle. Definition: a tissue cycle, it is called as a tricarboxylic acid cycle because the first compound formed in a cycle, it is with three carboxylic groups, so it is called as a tricarboxylic acid cycle. And this cycle is a series of enzyme catalyzed chemical reactions that forms a key part of aerobic respirations in a cell. This cycle is also called as a Kreb cycle or a citric acid cycle. The first product in this reaction formed is a citric acid, so it is also called as a citric acid cycle. Now the introduction: the citric acid cycle was discovered by the scientist Hans Krebs in 1937 and gave a name it as a tricarboxylic acid cycle. The scientist Hans Kreb received the Nobel Prize in Physiology or Medicine in 1953 for his discovery. The tissue cycle occupies a central position in metabolism and it meets most of a cell energy requirement. In a tissue cycle, here is a complete oxidation of acyl coenzyme A. The acyl coenzyme A get oxidized in a tissue cycle, and this acyl coenzyme A firstly form a pyruvic acid. The pyruvic acid is a product of the pyruvic acid is a last product in a glycolysis, and the pyruvic acid get converted into the acyl coenzyme A, and from that acyl coenzyme A the further cycle is ready. So it is a key product in a catabolism of carbohydrate, fatty acid, amino acids, and it get lead to formation of carbon dioxide and the chemical energy in form of GTP, that is a guanosine triphosphate. In short, in a tissue cycle, the acyl coenzyme A get catabolized into the CO2 and energy in form of GTP. This cycle provides precursors for a synthesis of amino acids, as well as it provides a reducing power in form of NADH, and this NADH is used in numerous other biochemical reactions. Thus, tissue cycle consumes acetate in form of acyl coenzyme A and water, and this reduces NAD to NADH and produces a CO2 as a waste product. The NADH generated by a tissue cycle is fed into a oxidative phosphorylation at time of respiration through an electron transport chain. In eukaryotic organism, the citric acid cycle occurs in a matrix of mitochondria. The pyruvate, we know that this pyruvate is a last product in a EMP pathway or a glycolysis, Here the one molecule of glucose is get converted into a 2 molecule of a pyruvic acid, and this is produced in a cytosol. After a production of the pyruvate, it get transported into a mitochondria or a matrix of a mitochondria through a transport protein, and the pyruvate molecule get dehydrogenated and also it get decarboxylated. Simultaneous, it leads to addition of a coenzyme A. The enzyme necessary for this overall reaction is a pyruvate dehydrogenase complex. This complex is having a number of roles, so it is called as a complex. This pyruvate dehydrogenase complex leads to formation of acetyl coenzyme A from a pyruvate. Here the raised hydrogen molecule from a pyruvate is accepted by a NAD, and the NAD is get converted into a NADH. Also the pyruvate get decarboxylated and one molecule of coenzyme A is added, so it leads to formation of acetyl coenzyme A. Now the schematic representation of a TCA cycle. The TCA cycle start from acetyl coenzyme A, but the first step in a TCA cycle is that the last product in a cycle is a oxaloacetic acid. This oxaloacetic acid get condensed with a acetyl coenzyme A, and this condensation is called as a Claisen condensation, where the methyl group of acetyl coenzyme A is converted into a methylene group in a citrate. The oxaloacetate and acetyl coenzyme A get condensed with each other and leads to formation of a citrate. The enzyme for this reaction is a citrate synthase, where this citrate synthase removes up the coenzyme A from a acetyl coenzyme A and forms a first product, that is a citric acid. Then the citric acid get dehydrated and rehydrated. Dehydration means removal of water molecule and rehydration means addition of water molecule. These are two reactions. Firstly, the citrate get dehydrated into the cis aconitase, and cis aconitate further rehydrated into a isocitrate. The enzyme for these two reactions is aconitase. The aconitase is playing a role for this conversion. Overall, the citrate get converted into a isocitrate. These are the isomers of each other. Citrate and the isocitrate are the isomers of each other, and here the intermediate is there, a cis aconitic acid. For this isomerization of a citrate into a isocitric acid, the enzyme necessary is a aconitase. Once the isocitrate is formed, it get oxidatively decarboxylated into the a compound, that is a alpha ketoglutarate. The isocitrate is get converted into a alpha ketoglutarate. The name of reaction is a oxidative decarboxylation. In this reaction or in this step, a OH group oxidized to a carbonyl, and in turn it facilitates a decarboxylation by stabilizing a carbanion formed on an adjacent carbon. Overall, in this reaction, a COOH group is removed, and a hydrogen atom which is removed from a isocitrate is accepted by a NAD and leads to formation of a NADH. Then next, the alpha ketoglutarate, further it get oxidatively decarboxylated into the product that is a succinic coenzyme A. In this reaction, CO2 group is removed from a alpha ketoglutarate. Also, a hydrogen molecule is removed from a alpha ketoglutarate, and the removed hydrogen is extracted by a NAD molecule, and this NAD is get converted into a NADH. Simultaneously, there is a addition of a coenzyme A with a SH group, and overall the alpha ketoglutarate, because of oxidative decarboxylation and addition of coenzyme A, get formed into a succinyl coenzyme A. The enzyme that catalyzes this reaction is a alpha ketoglutarate dehydrogenase complex. Alpha ketoglutarate dehydrogenase complex. The formed succinyl coenzyme A, it get converted into a succinic acid, and the enzyme synthesizing this reaction is a succinyl coenzyme A synthetase, and this is an example of substrate level phosphorylation. In this substrate level phosphorylation, the thioester bond that is present in a succinyl coenzyme A, this thioester bond is get converted into a phospho unhydride bond of a GTP or a ATP. So, here is a production of a GTP molecule at a conversion of succinyl coenzyme A to the succinic acid. The COA-SH group is removed, and the thioester bond present in it is converted into a phospho unhydride bond of a GTP. The enzyme necessary for this reaction is a succinyl coenzyme A synthetase. The succinic acid is formed. Further, this succinic acid get dehydrogenated into the fumaric acid, and the removed hydrogen is accepted by a FAD, that is a flavin adenine dinucleotide. This FAD is get converted into a FADH. So, in this reaction, succinic acid get converted into a fumaric acid. The enzyme that catalyzes this reaction is a succinate dehydrogenase. Further, the fumaric acid or a fumarate, it get hydrated. The addition of a water molecule across a double bond induces or introduces a OH group, and the fumarate get converted into a malic acid. The enzyme that catalyzes this reaction is a fumarase. So, the fumaric acid get converted into a malic acid through the reaction called as a hydration. Then next, the malic acid, with the help of an enzyme malate dehydrogenase, get converted into oxaloacetic acid, and this oxaloacetic acid is ready for a glycine condensation with a acetyl coenzyme A and formation of a citrate, and the cycle runs once again. Now, in detail, the reactions of a tissue cycle or a citric acid cycle. The first, here is the name of enzyme, that is a citrate synthase. This citrate synthase, it forms a citric acid by a condensation of oxaloacetic acid and a acetyl coenzyme A. It remove of the COA-SH group and adds acyl coenzyme in oxaloacetic acid. Aconitase, this enzyme catalyzes a isomerization of citric acid to a isocitrate. Once it removes up a water molecule and adds a water molecules immediately. So the isomer of citric acid is formed, that is a isocitrate. The enzyme is a aconitase. Then next, isocitrate dehydrogenase. This isocitrate dehydrogenase, it carry out a first oxidation step of a Kreb cycle, where the isocitric acid is oxidatively decarboxylated to a alpha ketoglutarate acid, and because of this dehydrogenation, a removed hydrogen molecule from a isocitrate is accepted by a NAD, and this NAD is get converted into a NADH. Next, alpha ketoglutarate dehydrogenase. This is a complex enzyme, and this having a different enzymatic activity resembling to the pyruvate dehydrogenase complex. This enzyme is catalyzing a reaction, that is a oxidative decarboxylation of alpha ketoglutarate, and because of oxidative decarboxylation of alpha ketoglutarate, it is get converted into a succinic coenzyme A. This enzyme, it is playing a different enzymatic role. It adds a coenzyme A to the alpha ketoglutarate and removes up a hydrogen molecule from it. So NAD is get converted into a NADH. The next, the role of succinic coenzyme A synthetase. The succinic coenzyme A, like acyl coenzyme A, has a thioester bond. This thioester bond is with a very negative free energy of hydrolysis. So this energy present in a thioester bond is converted into a phospho unhydride bond in a GTP. So the succinic coenzyme A get converted into a succinic acid that removes a COA group from a succinic coenzyme A and leads to formation of a GTP, and this is an example of a substrate level phosphorylation. The next, succinate dehydrogenase. This succinate dehydrogenase enzyme carry out a oxidation of succinic acid to a fumaric acid. So the removed hydrogen molecule is accepted by a FAD and it get converted into a FADH. And the succinate dehydrogenase, it is a only enzyme of a citric acid cycle that is tightly bound to the inner mitochondrial membranes. Other enzymes are present in a matrix of a mitochondria where this succinate dehydrogenase is tightly bound to the inner mitochondrial membrane. And this enzyme succinate dehydrogenase is a FAD dependent enzyme, flavin adenine dinucleotide dependent enzyme. The next fumarase, the fumaric acid reacts with a molecule of water and it get converted into a malic acid. Malate dehydrogenase, the oxidation of malate to a oxaloacetate, where because of oxidation of malic acid, one hydrogen is released. This is accepted by a NAD, and this NAD get converted into a NADH. Now the significance of TC cycle. Number of intermediate compounds are formed during a Kreb cycle, and these are used for a synthesis of biomolecules like amino acids, nucleotides, chlorophylls and cytochromes. We know that the one intermediate formed in a tissue cycle is a alpha ketoglutarate. This alpha ketoglutarate is a precursor for synthesis of glutamine and glutamic acid. So, this is a precursor for these amino acids. The succinyl coenzyme A or succinic acid, these two intermediates forms a heme group of a chlorophyll. Intermediate like succinyl coenzyme A takes part in the formation of a chlorophyll. The amino acids are formed from alpha ketoglutarate acid, pyruvic acid and oxaloacetic acid. A Kreb cycle releases plenty of energy required for various metabolic activity of a cell. By this cycle, a carbon skeleton are got, which are used in a process of growth and for maintaining the cells. Overall gain of energy in the tissue cycle is ATP generation is 12 ATP. You know that 3 NADH, that is a reducing power, are produced in this cycle. So 1 NAD is equal to 3 ATP and 3 NAD is equal to 9 ATP. 1 FADH is equal to 2 ATP and 1 GTP is equal to 1 ATP. Overall 12 ATP molecules are formed after one cycle of TC cycle or one completion of a cycle. So thank you very much. If you like my video, please subscribe my channel, share it to your friends and press the bell icon so that you get notifications.